The overall goal of the following experiment is to quantify mitogen-activated protein expression at the single-cell level. To achieve this, a yeast strain bearing the fluorescent expression reporter quadruple venous, controlled by the STL1 promoter and specific to the activation of the HOG1 MAP kinase, was generated. Fluorescent protein expression can be quantified either by a live cell microscopy assay, in which the cells are stressed directly on the microscope to facilitate real-time apparition of the fluorescence, or by flow cytometry, in which case the cells are stressed in a microtube and the protein synthesis is blocked at a given time by the addition of cyclohexamide. Only a few cells at a time can be analyzed with live cell microscopy, but the fate of the individual cells can be directly observed and correlated with other cellular properties. Flow cytometry, on the other hand, also allows assessment of mitogen-activated protein expression on large numbers of cells at once.
This method can help answer key questions in the signaling field, such as which proteins are involved in the regulation of gene expression.
Though this method can provide insight into the yeast HOG pathway, it can also be applied to other signaling cascades, depending on the availability of the proper expression reporter.
To prepare the cells for live cell microscopy, begin by inoculating 5 ml of synthetic medium with yeast and then grow the cells at 30 degrees Celsius overnight. The next morning, measure the OD600 of the overnight culture and then dilute the overnight culture in 5 ml of synthetic medium to an OD600 of 0.05. Grow the diluted culture at 30 degrees Celsius for at least 4 more hours. Next, filter about 200 microliters of freshly prepared concanavalin A solution into the well.